everyone, my name is Katharina Leeb, and today I'm going to talk about the drivers of behavior and movement of the endangered undulate ray, Raya undulata. During this last month, we all experienced our own behavior and movement determines our vulnerability, and just the same is true for marine populations and species, just for very different reasons. Knowing the spatial ecology is essential to assess the vulnerability to anthropogenic but also environmental pressures, and in the end also to assess the efficiency of potential protection measures. For Elasmobranchs that are K-strategy species and have a slow growth, a low fecundity and late maturity age, and are thus especially vulnerable to exploitation, there's simply a lack of information on their biology and ecology, and thus it makes it very difficult to assess the efficiency of protection measures. The undulate ray is a commercial species in the northwest of Spain, in Galicia, and is classified on a European level as endangered. And there have been few studies that assess their spatial distribution, and some that also try to identify the environmental drivers. But most of them use mark recapture methods, so they are restricted to certain temporal scales. And our study aims to identify biological and environmental drivers for the movement and behavior patterns of Raya undulata across different, also smaller temporal scales. Our study site is located in the northwest of Spain, in Galicia, more precisely in the Sierras archipelago, which you can see on the picture. And on the left map, you can see that around these islands, the black box designs the borders of the partial MPA that is placed. In this MPA, there is marine protected area. No recreational fishing is allowed, but commercial fishing is allowed with some restrictions. So our study area was right in between these islands, where we had a passive acoustic telemetry array to detect the movement of the rays. In total, we had 20 receivers with overlapping ranges, so that from the detections, we could later calculate positions. And in the yellow area, you can see the whole area where we were able to get positions of our individuals. We also deployed two reference tags, V13 and V16, in order to make sure that the detections that we get and the patterns that the movement patterns that we detect are due to the actual movements of the rays and not just to environmental variations. We also monitored the temperature in this area with a temperature data logger, which you can also see on the map. And all the individuals were tagged in a place where accumulations of the undulate ray have been previously observed. In total, we tagged 44 individuals, 21 females and 23 males, and they were tracked from June to December 2019, last year. So we had a total period of 6 months. Apart from the acoustic telemetry positions, we also took biological data. So we determined the sex and the body size while we were tagging. And as already said, we also monitored the temperature in situ and included an index for the upwelling conditions in our study, which is called upwelling index, and which is estimated by the Spanish Oceanographic Institute based on the meteorological conditions that either favor upwelling or downwelling conditions. We had to perform several data processing steps because our array had some technical restrictions. So our skates would not be detected when they were moving in between rocks and also when they were buried in the sand. We tagged our skates internally in their peritoneal cavity, which is in the lower part of the body, and when they are buried in the sand, it is very likely that the transmission does not get to the receivers. How did we do this filtering? We calculated trajectories from our positions that were characterized by the distance and the time between the relocations, and we used these distance and times as filter criteria in order to assure that we only include movements inside the study area in our analysis, and that we identify periods where we had to interpolate the data, some data. For the data analysis, we used generalized additive mixed models in order to identify the drivers for the movement. We estimated 3 metrics for the movement. One was the probability of presence, the other one the weekly home range during day and during night, which was defined as the 95% kernel utilization distribution, and the speed, which was defined by the distance divided by the time between 2 relocations. These movement estimators were included as response variables in our model, whereas the biological and environmental factors such as sex, the disk length, temperature and upwelling index were included as explanatory variables. We also included random effect for the temporal autocorrelation and the autocorrelation within individuals. We then performed a stepwise backward model selection in order to identify the drivers. This led to the following results. We found clear seasonal and diel patterns, that males are more active than females. We found no effect of body size and only very small effect of the environmental variables. Let's have a look at these results more in details. For the seasonal patterns, it's important to say that we only had a data set of 6 months, which allows us only to analyze summer and autumn. But within these 2 months, we found very clear patterns. Here you can see the prediction plots of the optimal fitted models. On the very left for the probability of presence, which is first slightly decreasing and then more drastically decreasing from the middle of the study period, so from autumn on. Also the home range area, the space use, is almost linearly decreasing towards the end of the year. And the activity is shifting from lower levels to higher levels from the beginning towards the end of the study period. Because we only have half of a year, it would not be we could argue that also the probability of presence is decreasing because maybe the dry undulata are fished. But during this time of the year, fisheries were closed, and now we have additional data for the rest of the year, and actually 25 of the individuals came back. These seasonal patterns indicate that our study area loses in importance for the species with the year. Apart from these seasonal patterns, we also found very clear diel patterns, which is Diel Cycle, with high activity levels during midnight and low activity levels during the day. On the left, you can see the speed over the hour of the day, and the blue shaded areas is the nighttime. With these low activity levels, also the space use, the area that they use, decreases during the day. And if we have a closer look now at the activity levels, here in this animation you can see the movement of all the tagged species on the 14th of July. And you can see that at certain point at 10 o'clock in the morning, they stop moving, and in the afternoon they start moving again. And it's very impressive with which synchronicity this happens. So now at 10, they stop moving, and 3, they start again. Also, a closer look at the space use. Here on the left side, you can see the total home range areas for 2 individuals during the day and during the night. And you can see that not only the size of the area changes between day and night, where during the day is smaller, but also the shape of the area. Coming now to the biological drivers, we only found an effect of sex on activity. So males are moving 30% faster than females, and we found no effect of body size. This might be surprising because body size is a driver for different behaviors that actually should reflect in activity levels, but it is important to note that we only detected rather large individuals and in a small size range, which might be the reason why we did not find an effect of body size. Another interesting thing is that if you compare the size of our individuals to the size at maturity, you find that only a few of the female individuals are actually considered as mature, where over half of the male ones is mature. So the difference in activity that you can see might also not only be due to sex, but to maturity status and the corresponding behaviors. The environmental drivers had a very, very small effect on presence and activity. The upwelling had a slightly negative effect on the presence of probability. So if you have a high upwelling index, it means favorable upwelling conditions. If you have a low one, it means favorable downwelling conditions. So with favorable upwelling conditions, you would have a lower probability of presence, but a higher activity. And for the temperature, also you have a higher activity at higher temperatures. The problem with these environmental variables that we included in our study is that for once the upwelling index does not correspond to only to our study area, but to the whole Rías Baixas region, which is in the south of Galicia. Plus, although the upwelling conditions favorable upwelling conditions lead to primary production and can be interpreted as food ability, in our study we did not take into consideration the time lag that occurs between upwelling conditions and primary production or food availability. Also, the sea temperature only has an effect on activity and not on the presence, or the Diel cycle, which makes it unlikely that it is related to diel or seasonal patterns, and rather to hydrodynamic processes, just as the upwelling index. So to really assess the environmental drivers for the movement of the species, it would be essential to assess these variables and factors also in situ. In our study period. This leads already to our conclusions. We found that the seasonal and diel cycles, which determine the presence, but also the activity of the species and the space they use in our study area, might affect the vulnerability. Because, for example, if the individuals are moving more, they might be more likely to encounter static fishing gear, which in our case would be allowed in the area. Same is true for males that move more than females, which additionally, if they are removed from the population, leads to a change in the demographic composition of the population. It is important to also assess the underlying biological traits, because it makes a big difference if the study area is, for example, a reproduction ground, a mating ground, or if the observed activity levels correspond to foraging. There's further studies that include maturity status and different ranges of sites, as well as a longer study period, would be essential. And as already said, the environmental drivers need further assessment. To conclude, a potentially small MPA could work, as we found small home range areas that at the same time take into account the temporal variations in activity and presence. Thank you for your attention, everyone that is watching, everyone that is in the room, and another thank you to my supervisors that supported me throughout the whole process. Thank you.